Okay, so merhaba guys. My name is David and I'm PhD student from University of Ostrava, from Czech Republic. And I'm going to tell you something about evolution and unprecedented variants of genetic code in one group of algae. Let me just change this thing. Okay, so you all know the standard genetic code, which is basically the set of rules how are triplets of nucleotides translated into amino acids. The standard genetic code is used by majority of organisms, and we are also using it in our nuclear genomes. But some organisms can use so-called alternative genetic codes, where some codons are used in different way than in standard genetic code. And for eukaryotes, it's usually UAR, which means UAA, and UAG, which are stop codons to encode glutamine. And in bacteria, the most common reassignment is UGA to encode tryptophan. And what you can see here is that majority of those codon reassignments are connected to stop codons. In eukaryotes, we can find also sense-to-sense reassignment in case of yeasts. And you can see it here, where CUG codon, which normally encode for leucine, is used as serine or alanine. And in our lab, we are mainly focused on nuclear genetic codes, but today I will tell you something about mitochondrial genetic codes, because we can find a lot of sense-to-sense codon reassignments there, as well as stop-to-sense. When people sequence some new organisms, they usually don't care about genetic code at all, and they translate it according to what they can find on NCBI. So we kind of assumed that they missed especially sense-to-sense codon reassignments, because they are not really visible when you do BLAST searches or when you prepare multiple sequence alignments. You don't see in-frame stop codons as in case of stop codons. Yeah, and I would like to apologize that my camera is placed so weirdly. It's in the left bottom corner of my laptop. And I don't know why it's so stupid. So I just want to apologize for that. So at the beginning of our project, we were thinking that people missed a lot of interesting genetic codes, and all those and a lot of predicted proteins on GenBank are translated with wrong genetic code. So we checked the literature and we found one paper from 2017 where Emmanuel Noutahi, I hope that I pronounced his name well, and other people, they created a software called Core Tracker, and with this software they checked genetic code of 40 metazoans and more than 100 yeast mitochondrial genomes. And they found some interesting things there. But if you consider the eukaryotic diversity, many people usually think about eukaryotes just as land plants. I will mark those branches, land plants here, metazoa, and fungi. But people tend to forget that the majority of eukaryotic diversity is in unicellular organisms. So what we decided was to check if there is something interesting in mitochondria of these unicellular eukaryotic organisms. And our pipeline was super easy what we did, we collected all jumping files from of mitochondrial genomes, except those from metazoa and yeast. We sorted predicted proteins from those genomes to separate multiple sequence alignments, and then we check all conserved positions, which means where some amino acid was conserved in more than 70% of organisms, and we collected information about underlying codons for all organisms. And here is one example to make it more clear. So in case of Attractomorpha, which is unicellular algae, what you can see that in position with conserved leucine here, this unicellular algae has CGG, which usually, or in standard genetic code, it encodes arginine. So when we collected information of all positions where Attractomorpha has this CGG, we found that there is no single position with conserved arginine, but the most occurrent amino acid was leucine. And now there is no need to care about this blue bar and purple bar, but only this red bar. So we can see a strong signal here for CGG codon to encode leucine. So when we did this thing for more than 1,000 organisms from GenBank, we found one group of algae which seemed particularly interesting, and this group of algae is called Sphaeropleales. And we found then in this group there are some organisms which are using UAG to encode leucine, UAG to encode alanine, AGG, alanine, AGM, methionine, AGA, alanine, and CGG leucine. In the first two cases, it's the example of stop to sense codon reassignment, but in case of AGG and AGA, those codons normally encode arginine as well as CGG. But it seemed that in case of this unicellular algae, they encode different amino acids. So what we did first, we checked the literature, what we know about this group. And in 1996, there was a paper where these probably Japanese guys, I assume, presented a paper when they claimed that UAG probably encodes leucine in one group of Sphaeropleales. Leucine, I'm sorry for my drawing, guys. And some other guys from this group use UAG to encode alanine. So, alanine, sorry for my pronunciation. So that was from 1996. But in 2014, there was a paper where they claimed that organisms which belongs to Hydrodictyaceae are using UAG to encode leucine, as we can see in Scenet desmatce. So if we go back here, it was known before our research, let's say, that UAG encodes leucine in this group. This codon reassignment was kind of controversial, let's say, and all those other codon reassignments, they will be completely new, and some of them unprecedented, which means basically that you cannot find them in any other organisms. So What we did basically, as I described our method at the beginning, that we created multiple sequence alignments and checked those conserved positions. What we did next, we collected Sphaeropleales, mitogenomes from Sphaeropleales from other databases than just GenBank. So we use like all publicly available data from many papers and many people. And when we look at the predicted genetic code, which we predicted, we saw that there is no codon reassignment which would be present in more than 70% of those organisms. So we run our analysis again with the genetic changes which we predicted with those eukaryotic data set, this big data set, and we apply them to this group separately to get more conserved positions. So then the signal for genetic code was genetic code reassignment or genetic code changes was higher. Let's say what you can see with this blue and purple bar. The purple bar actually is the second iteration of the procedure using only organisms from Sphaeropleales. I hope that is at least a bit understandable. Okay, so what we can see here is that genetic code in this group of algae is very, very complex, and AGG, which normally encodes arginine, can encode methionine here, or alanine. In case of AGA, it seems that it can encode alanine and not canonical arginine. And in the case of UAG, which normally stands for stop codon, we can see that they are using it as a leucine, some of them, these groups, tRNAs, or alanine in case of Hydrodictyaceae. So even from this table, you can see that we kind of support this paper from 1996 in case of UAG stop codon. But all those findings, let's say, they are based solely on multiple sequence alignment, and that's not like it's only one evidence, and it's weak evidence. Let's say it's not that strong. So what we did next, we check if there are some tRNAs which would be able to support our hypothesis, and if I would clean everything what's here, undo OK. Those organisms which are depicted in bold, it means that we have full mitochondrial genomes for those, and others we have for them we have only sequences for protein coding genes. So we were not able to check the tRNAs there. But for those organisms depicted in bold, when we check their genomes for the tRNAs, if there are such tRNAs, like canonical tRNAs for AGG to encode arginine, or AGA to encode arginine, and those other changes, we never found any canonical tRNAs. But what we found instead is that, for example, in case of Chromochloris, I'm sorry guys, in case of Chromochloris zophingensis, you can see that we predicted that AGG is used as methionine and CGG as leucine. And we found two non-canonical tRNAs in this organism, and one of them with anticodon cognate to AGG, which clusters with tRNAs for methionine. And that's exactly what we predicted. And we can see the same case here with CGG to encode leucine. So we predicted that this organism is using CGG as leucine CGG, and we found tRNA with anticodon CCG, and this tRNA cluster with other canonical tRNAs for leucine. And when we check other reassignments with the same approach, we found that in case of this UAG, you cannot see my mouse, guys, I'm sorry. In case of UAG, here we predicted that it encodes leucine, and when we check if there are some tRNAs which would support this hypothesis, we found them, and they nicely cluster with other tRNAs, canonical tRNAs for leucine. Little bit more complicated case was here, all these organisms which are using AGG as alanine. We didn't find canonical tRNAs for arginine there, but we found some paired tRNAs which clustered with tryptophan, and that was unexpected, but the signal in those organisms to encode alanine is so strong, so we are expecting that those tRNAs, they emerged via duplication of canonical tRNA for tryptophan, and then this tRNA, thanks to neofunctionalization, kind of changed its specificity. So the last case are those tRNAs which can recognize UAG and encodes alanine, and we found them, and those nicely cluster with canonical tRNAs for alanine. So as we expected. Based on those searches, we prepared this scheme where we describe evolution of mitochondrial genetic code in this group, basically. And what's interesting there is that it seems that majority of those codon reassignment happened according to codon caption theory, which basically says that at the beginning, the organism kind of stopped to use some codon, so it disappears from the genome, and you can see that those codons which were changed are kind of decreased at the beginning of the speciation of this green algae, and they can totally disappear from the genome. And even if I would go back, you can see that those codons are usually not there, as we can see here, or they are reassigned to something in majority of those cases, or they are there in very, very low numbers. What's interesting about evolution here is that this AGG to some other amino acid than arginine happened at least 3 times in the evolution of this group, and we assume that according to phylogenetic tree of those tRNAs and the phylogeny of those organisms. So that's an interesting thing. But what's even more interesting for me is that people knew, it was proposed before, that in Sphaeropleales there are not just codon reassignments in a way that stop codons or sense codons are reassigned to encode something different, but it was proposed, I think, in 2014 that this group of algae, some of them are using alternative stop codons, which means that they are not using only UAA, UAG, or UGA as a stop codons, but they are using also UCA or UCG, and those codons normally stands for serine in the standard genetic code. So when I look at the genetic code of those organisms, it immediately came to my mind that I will find only mitochondrial release factor 2, because they are using UAG to encode some amino acid. And as you maybe know, in bacteria, plastids, and mitochondria, we can find two release factors, which are those proteins which basically mediate the termination of translation and release of the peptide. So you can find two of those in plastids, mitochondria, and bacteria. Mitochondrial release factor 1 can recognize UAA, stop codon, and UAG. And mitochondrial release factor 2 is able to recognize UAA as well, and UGA. So according to the fact that UAG is used to encode an amino acid in this group of algae, I kind of expected to find only release factor 2 there, but it was the opposite actually. So what we found there were only mitochondrial release factor 1. So when we were thinking about it, we knew that those proteins has to be somehow weird in the part of the protein which is responsible for recognition of a stop codon. So what we did, we collected all possible data to check release factors from this group, because usually people sequence only plastids or mitochondrial genomes, so we didn't have so many sequences, but we found, I don't know, it's like 10 or more sequences of release factors, because they are not nuclear encoded but they are encoded they are not encoded in mitochondria, but they are in the nuclear genomes. And when we check them, it looked very, very, very interesting, because as I said before, mitochondrial release factor 1 should not be able to recognize UAG, no, it is able to recognize UAG, and the position which is critical for recognition of that guanine base at third position is this glutamine 181 here. And what you can see is that in some Sphaeropleales, that glutamine is mutated to some different amino acid, to threonine. And here, what we can see as well is that it's only shared between Hydrodictyaceae, Neochloridacea, and Cyanidesmaceae. And if we would go back and check this table once again, you will see this table a lot. Those groups are exactly those groups where you can see stop codon UAG to be reassigned to encode some amino acid. So that was very interesting. but it's not all, because we also found that in some organisms there is mutated position 186, which is responsible for the discrimination of adenine against guanine in the second position of a stop codon. So that's why release factor 1 is not able to read UGA. And what we found here is that it's mutated in some organisms, again, together, interestingly, together with position 206. You can see conserved serine here, and those two residues are typical for release factor 2. And when we check those organisms, what stop codons they are using in 13 conserved protein coding genes, some of those organisms where we found those mutations, they are able to use UGA stop codon. So it basically seems that mitochondrial release factor 1 in this group of algae is kind of able to mimic behavior of release factor 2. And that's really interesting. And the last thing, what probably never, nobody asked ever before, is how is it possible that this release factor is able to recognize alternative stop codons? It's very difficult to answer such questions based just on primary structure of this protein. But since we were lucky kind of to see those reassignments with nicely matches with the findings what we had, we kind of look if there is something specific for this group of algae, because nobody else is able to use UCA and UAG as a stop codon. At least we don't know such organisms. And what we found is that in position 182, all organisms across tree of life, all of those organisms are using arginine in this position. But in case of Sphaeropleales, they are not using arginine there. So we kind of we suggest that this different amino acid than arginine in this position is probably responsible for the ability of release factor 1 in Sphaeropleales to recognize cytosine in the second position of a stop codon. So what you can see is that it's super weird to talk to nobody. I'm sorry. I mean, I cannot see anybody. That's what I meant. I'm sorry. What you can see is that there is a lot of changes in genetic code in this algae. And it's really interesting, and you wouldn't probably find any other organisms with genetic code so complex in their mitochondrial genomes. And it was so interesting that one day when we were finishing our paper, my boss called me and asked me to sit down and send me this beautiful paper written by some guys from Canada. And it was very interesting to read it, because they basically found 80% of things what we found, and they published it in MBE, and that's exactly where we wanted to send our paper. It was really interesting to see that they had the same conclusions as we had. And I have to admit that I never have so many interesting emotions while reading some paper. So we had to do something about it. You know, I don't want to say that we got scooped, but it's kind of, you know. So we were super close to publish our paper, but some guys were just faster and they published it. And we didn't want to check their paper like a lot because we didn't want to use what they used. We didn't want to steal from them. So we just like quickly checked the figures, what they had, if it's really the same thing as we did. And then we decided that we will finish our paper, and at the end we will check their paper, and we will add one section when we will compare those papers. And that's exactly what we did. In case of this Canadian group, they found some codon reassignments, what we found, but they missed some. So, but they also add something new. So, because their sampling, they checked more organisms, not just the Sphaeropleales, but they, for example, found that Pycnococcus, which is different algae, use AUA to encode methionine. But what they totally missed is this AGA reassignment from arginine to alanine, basically because they didn't have the data. We spent a lot of time collecting our data set, and we were predicting all of those proteins from genomes, and it took us a lot of time. So at the end, I was happy that at least we found something what they didn't find. But they published the MBE and we didn't. So that said, okay, so they didn't find this AGA. And the other thing is they didn't ask how is it possible that Sphaeropleales are able to recognize UAG. Because if you want to use this stop codon, there has to be something weird about release factor. And they didn't also ask, about those alternative stop codons, and we did. So that's also some additional thing what we had in our paper. But they did a great job with tRNAs, and they have amazing figures. It's a very nice paper, and they did some analysis what we didn't. So it's definitely good to check also their paper if you are interested. At the beginning of this project, they were predicted only one genetic code for Sphaeropleales, and it was genetic table 22, which says that UCA is used as a stop codon, and UAG, which normally encodes stop codon, is used as leucine, and it was predicted as only one genetic code for this group of algae. Nobody from this group is using this genetic code, which is really interesting. And we had to define 8 new genetic codes, so people would be able to translate proteins from mitogenomes of this group correctly. I'm sorry, I stuttered a bit. So you have to keep in mind that everything, all those proteins on GenBank, those are just predictions. And people, they very often they don't care about genetic code, and I don't blame them. I don't blame them in case of sense-to-sense, because it's very difficult to spot these changes. But sometimes when people see that there is some stop codon in frame stop codon, they just select some genetic code which is available on NCBI and use this one, and they don't care. So there is a lot of mistakes in protein prediction just because people don't care. And the beautiful thing, so as I said, we We described 8 new types of genetic code, but it's not clear that it's, I don't want to say easy, but it's that easy. But we don't know what's going on with AUA. Codon, which normally stands for isoleucine, but we don't have basically any data or very few conserved positions for Sphaeropleales in this case, and this codon is very often reassigned to methionine. So maybe these predictions will be changed in future. And at the end, our paper was not published in MBE, but in GBE, which has way lower impact factor than MBE. But you know, the purple color of the title is very nice, better than the blue one in case of MBE. And they also, together with this paper, they published a highlight on our paper. And when they told that to my boss, he immediately insisted that they have to include also this Canadian group. So in the highlight, thank you guys. In the highlight, you can read about both of those papers. Yeah, so this is the end. I hope that it was at least a bit understandable. I have to confess that this is my first webinar, and it's super weird when you cannot see anybody and you are just talking to your camera and nobody's there. So I would like to, at the end, thank to my amazing supervisor, Margaliaš. I would like to thank to Czech Grant Agency for financial support. I would also like to teşekkürler to my great arkadaşım Ezgi Altınışık, the Twitter'ını Kraliçesi. I hope I said the queen of Twitter. and RSG Turkey as well for making this webinar. And I would also like to thank you for your attention. And at the end, I would like to invite you for Ankapai next year, which will be happening next year, I hope, which will happen next year. It's going to happen next year, whatever. It's a Python workshop where we are basically teaching basics of Python and also Python for bioinformatics and how to use BioPython, for example, how to work with blast results and many useful things. So definitely check RSG Turkey Twitter and Ezgi's Twitter, because there will be definitely something happening next year. I hope that it will be possible to fly it. And yeah, thank you for your attention again. And now I don't know what to do, but I think it's time for questions. Okay, so the first question is not about my presentation, but it's about the workshop. So the question is, I'm interested in this workshop of Python. Can you send the link with more information? Now there is only GitHub page, and yes, I will. I think that this video will go to YouTube or we have your email or something because you registered for this webinar. So I will provide you the link for GitHub page where you can basically download all the materials for the workshop, what you need, and you can start to do it yourself, or you can wait for next year when we will be talking about everything in, I hope, Istanbul or Ankara. I don't know. Last year was Ankapai, but next year it can be Istanbul. We will see. But maybe Ankara is better because it's in the middle so people from different parts of Turkey can like visit it easily. Okay, so do we have more questions? Yeah, I'm afraid that we don't, and I'm also afraid that it was because it was not really understandable and maybe too fast. I didn't expect that I wouldn't be able to point to things so easily, so I made those horrible drawings there. Okay, so there is one more question. Okay, so what happens if you transfer any gene from an organism with alternative genetic codes to another organism? This is very interesting question because of many reasons. The first reason is like that maybe you touched why in some organisms it's maybe even beneficial to use alternative genetic code. Maybe it can be some, some protection against viruses, maybe, or some other organism. Well, probably viruses mainly. So, if you would take the gene with alternative genetic code, especially like if there would be sense-to-sense reassignment, it should be okay, probably. It depends of the specific amino acid mutated, but not mutated, like specific amino acid changed. But in case of stop codons, you would not be able to make a functional protein. And interestingly, this is a strategy of one virus of bacteria, I think. I hope that I remember it correctly. There is a virus which is virus of one bacteria with alternative genetic code, and this bacteria is using UGA to encode tryptophan, and this virus has in its genome gene for release factor 2, and at some point the virus start to express this release factor 2, which means that all proteins from the bacteria are just are not translated correctly. So that's I think that this is right. So it can also be used in those arms race, let's say. And there is one lab in Czech Republic, in České Budějovice, and they are currently trying to use one organism with alternative genetic code as a model organism and just test some things there, because it's really interesting in many ways. Okay. I think that there are no more questions. I think that's it. Thank you guys. Thank you again for your attention. And I think that's it. So, Kurushurus. Yeah, that's correct. Okay. Bye-bye, guys. I don't know how to stop it.